Hello guys, welcome to this lesson. We're going to talk about protein secondary structures. But before starting, I want to tell you what we're about to learn. We will make an introduction to protein secondary structures. Then we will talk about the two most famous secondary structures, alpha helices and beta sheets. Afterwards, we will talk about non-periodic secondary structures, which are turns and loops. And finally, we will discuss self-assembling peptides, and we will analyze a study case, EAK162. So let's get started. In the world of proteins, the secondary structure refers to the local folding patterns and spatial arrangements of amino acid residues within a polypeptide chain. While the primary structure outlines the linear sequence of amino acids, the secondary structure delves into the immediate, recurring spatial motifs adopted by the protein's backbone. The two most common types of secondary structures are alpha helices, characterized by a coil and helical structure, and beta sheets, where are segments of the polypeptides line side by side, forming a sheet-like arrangement. The alpha helix is a prevalent and essential secondary structure found in proteins, characterized by a tightly coiled helical arrangement of the polypeptide chain. This structural motif is stabilized by intramolecular hydrogen bonds formed between the carbonyl oxygen of one amino acid residue and the amide hydrogen of an amino acid residue 4 positions away. This regular pattern of hydrogen bonding imparts stability to the helical structure. The alpha helix often resembles a spiral staircase, with the side chain of the amino acids extending outward from the helical axis. Let's now see some key features of the alpha helix. So it includes stability. The hydrogen bonding, in fact, imparts considerable stability to the alpha helix, making it a common and robust secondary structure in protein. Then we have that it is right-handed. The helix is right-handed. In fact, it means that if you were to move along the helix as if waking up a staircase, the helix would spiral to the right. Another feature is its helical turn. The standard alpha helical turn involves 3.6 amino acid residues per turn. Then pitch and diameter. So the pitch, which is the vertical distance between turns, and diameter of the alpha helix are relatively uniform, contributing to its regular and compact structure. Let's see now some common occurrences. So alpha helices are frequently found in protein structures and are often associated with regions that traverse a cell membrane or play a role in structural stabilization. And their protein function, well, we need to see that the alpha helix is involved in various functions, including forming transmembrane domains in membrane proteins, participating in protein-protein interactions, and contributing to the overall tertiary structure of a protein. Notable examples of proteins containing alpha helices include myoglobin, a protein involved in oxygen storage in muscles, and alpha helix-rich keratin proteins found in hair and nails. On the other hand, the beta sheet is a fundamental structural motif in proteins. In a beta sheet, segments of the polypeptide chain adopt an extended and relatively flat conformation, arranged side by side, resemble a pleated sheet. This structure is stabilized by hydrogen bonds formed between the amide group the amine hydrogen of one peptide bond and the carbonyl oxygen of another, creating a network of hydrogen bonds that run perpendicular to the direction of the polypeptide chain. There are two main types of this: parallel and antiparallel. In a parallel beta sheet, adjacent polypeptide strands run in the same direction, while in an antiparallel one, adjacent strands run in opposite directions. The choice between parallel and antiparallel configurations significantly influence the stability and properties of the beta sheet. Beta sheets are crucial for the structural integrity of proteins and contribute to their overall stability. They often form the core structure of protein domains and are frequently found in the interior of globular proteins. Beta sheets also play a crucial role in protein-protein interactions as well as in formation of larger, more complex protein structures. The arrangement of beta sheets in a protein, along with other secondary structures and the connecting loops, determines the protein's tertiary structure. Understanding the beta sheet content and its specific arrangement is essential for gaining insights into the function, stability, and folding of proteins, as well as for protein engineering and drug design in the field of molecular biology. There are other secondary structures fundamental for tertiary structures: turns and loops. In biochemistry, turns and loops refers to specific structural elements that are crucial for the proper folding and functioning of proteins. Let's start with turns. A turn is a type of secondary structure in a protein that involves a change in the direction of the polypeptide chain. Turns are characterized by a tight band or loop in the protein structure, and they often connect different secondary structure elements, such as helices and strands. Turns play a significant role in protein folding, stability, and functionality. The most common types of turns include beta turns type 1 and 2. Type 1 beta turns involves a 180-degree turn in the polypeptide chain and are characterized by a hydrogen bond between the first and fourth amino acids in the turn. Type 2 beta turns also have a 180-degree turn but form a hydrogen bond between the second and the fifth amino acids. Now let's talk about loops. Loops in the context of protein structure refer to regions where the polypeptide chain does not form regular secondary structures like alpha helices or beta strands. Loops are often found on the protein surface and are more flexible compared to structured secondary elements. They connect different secondary structures and can contribute to the overall flexibility and adaptability of a protein. Loops are essential for accommodating functional sites, allowing proteins to interact with other molecules and facilitating conformational changes. Now let's talk about self-assembling peptides. This is a breakthrough for biochemistry and is one of the most amazing types of topics to talk about. So self-assembling peptides, as I've told, are a fascinating class of molecules with the unique ability to spontaneously organize into well-defined structures without external guidance. These peptides are designed to exhibit a controlled and predictable self-assembly, driven by non-covalent interactions, such as hydrogen bonding, hydrophobic interactions, and electrostatic forces. The key feature of self-assembling peptides is their ability to form intricate nanostructures or supramolecular architectures, mimicking natural biological systems. Let's see now the design principles. So, self-assembling peptides are often engineered with specific amino acid sequences that promote spontaneous assembly. Common motifs include alternating hydrophobic and hydrophilic residues, which drive the formation of stable structures. Let's see some applications. In the case of the biomedical field, self-assembling peptides find extensive use in biomedicine, contributing to the development of drug delivery systems, tissue engineering scaffolds, and regenerative medicine. Their ability to mimic natural extracellular matrices make them valuable in creating biomimetic environments for cell growth and tissue repair. In the case of nanotechnology, self-assembly peptides serve as building blocks for constructing nanoscale materials and devices. These applications range from designing nanocarriers for drug delivery to creating nanoscale sensors and imaging agents. In the case of material science, furthermore, self-assembling peptides are employed in these cases to produce functional materials with tailored properties. Let's see some examples, like the development of nanofibers, hydrogels, and responsive materials for various technological applications. The mechanisms of self-assembly is another topic of interest. The self-assembly process typically involves a balance between attractive and repulsive forces among the peptide molecules. Hydrophobic interactions, hydrogen bonding, and electrostatic interactions guide the peptides to spontaneously organize into specific structures. Now let's see the peptide amphiphiles. It is a subclass of self-assembling peptides, and it possesses both hydrophilic and hydrophobic regions. This dual nature allows them to form organized structures, including micelles and nanofibers, in aqueous environments. Now let's see the biological inspiration. Self-assembling peptides draw inspiration from natural biological systems, where proteins and peptides self-assemble to perform specific functions. This biomimicry enhances their compatibility with biological systems and their potential for applications in medicine. However, of course, it will have some challenges, and of course, it will also have advances. So, in the case of challenges, it is mostly in designing self-assembling peptides, which includes achieving precise control over their structures and functions. In the case of advances, we see that advances in computational design and synthetic biology contribute to overcoming these challenges, allowing for the creation of increasingly sophisticated self-assembling peptide systems. An example of a self-assembly peptide is EAK16-2. It refers to a peptide sequence that exhibits self-assembling properties. Self-assembling is a process by which these molecules spontaneously organize and come together to form larger order structures without external guidance, as we have said. In the case of this molecule, the self-assembling behavior is attributed to its unique amino acid sequence. EAK16-2 is a design peptide that includes alternating hydrophobic and hydrophilic amino acids. This alternating pattern allows the peptide molecules to organize themselves in a specific way. The hydrophobic amino acids tend to cluster together to minimize exposure to water, while the hydrophilic hydrophilic amino acids interact favorably with water. This leads to the formation of organized structures, often in the form of beta sheet structures. These structures are stabilized by hydrogen bonding between adjacent peptide chains. The self-assembly of EAK16-2 can result in the formation of nanofibers or other higher-order structures, depending on the environmental conditions and specific properties of the peptide. The resulting structures have potential applications in various fields, including biomaterials, drug delivery, and tissue engineering. In summary, the self-assembly of EAK16-2 is driven by the inherent properties of its amino acid sequence. The hydrophobic and hydrophilic interactions between amino acids lead to the spontaneous formation of organized structures, showcasing the intriguing ability of certain peptides to assemble themselves into functional architectures. Apart from all we have explained, supersecondary structures, also known as motifs or folds, are intermediate level structures that describe patterns of secondary structure elements and their spatial arrangement within a protein. While primary structure refers to the linear sequence of amino acids and secondary structure describes local folding patterns, supersecondary structures provide insights into how these secondary structural elements are organized to form specific recurring patterns or motifs. Here are a few of supersecondary structures. So we have beta-alpha-beta motifs, BAB. These motifs consist of a beta strand followed by an alpha helix and then another beta strand. It is a common arrangement in proteins involved in ligand binding and enzymatic activity. Then we have as well the example of helix-turn-helix motif, HTH. Commonly found in DNA-binding proteins, this motif involves two alpha helices separated by a short turn. The helices work together to bind the specific DNA sequences. Well guys, that's all for this lesson. I hope you liked it. I see you in the next one. Bye bye.